Hi, I'm Dr. Naomi Moris, and I'm a group leader of the Stem Cell and Human Development Lab at the Crick Institute. We have a really exciting new position for a clinical PhD student to come and join our team, researching the effects of mutations and mosaicism in human embryonic development. As a lab, we're interested in figuring out how the early human embryo establishes the organization of the body plan at perigastrulation stages. Since there are ethical and technical limitations on using human embryos at this particular stage of development, we use pluripotent stem cells as an in vitro model system. In particular, we generate 3D human gastruloids, which are able to self-organize into structures that bear resemblance to a 20 to 22-day-old human embryo. They undergo spontaneous symmetry breaking, polarize their gene expression, and then undergo morphological changes, including axial elongation. As such, they're a great model to explore how cells make fate decisions in a coordinated manner over space and time. For this project, we've teamed up with some fabulous colleagues, Dr. Christina Diaz, a clinical geneticist, and Dr. James Turner, a senior group leader and geneticist, to explore how various genetic mutations observed across patient populations affect this particular time point in human development. In particular, we want to explore how epigenetic regulation mediates gastrulation and early organogenesis stages of development, and how mosaicism in the embryo might be fine-tuning the observed clinical effects. We have a brilliant bunch of diverse lab members who are driven and curious and highly creative, and we hope to find somebody equally motivated to join our team. We hope you'll apply.
Hi, I'm Christina Diaz. I'm a consultant clinical geneticist and clinician scientist at King's College London and collaborating with the Crick. And I study chromatin remodeling genes in the context of developmental disorders. Chromatin remodelers are protein complexes that regulate the accessibility of our genome in a developmental time point and cell type specific manner. Several of these proteins are involved in regulating perigastrulation, cell fate specification. And mutations in chromatin remodelers can cause severe developmental defects. For example, in the mouse knockout of one of these, the ARID1A gene, its development arrests at gastrulation with lineage specification defects, and the heterozygote knockout does not survive beyond mid to late gestation. In humans, mutations in this gene can cause cardiac, skeletal, craniofacial defects, and intellectual disability, such that most surviving patients have mutations that are less deleterious or mosaicism. Mosaicism means that the individual has different cell populations within their bodies, some with the mutation, others without. And we've also observed that in patient-derived induced pluripotent stem cells, we also see differentiation defects. So harnessing induced pluripotent stem cells derived from patients and cells with targeted mutations, we can start to explore the role of these proteins in early development and organogenesis, and explore how mosaicism can play a role in early development and survival. I'm very excited to have partnered with Drs. Naomi Moris and James Turner to start to explore these questions. There's vast experience in developmental biology, and the experimental methods that Dr. Naomi Moris has developed will be central to this project. Their experience, synergized with my interest in human development and disease and the multidisciplinary environment that the Crick has to offer, have converged on what I believe is a very exciting project that really transverses basic human biology and clinical outcomes. I'm really looking forward to supporting a clinical PhD student taking this leap into science through this project that stemmed from clinical observations to address fundamental questions on human development and developmental defects. We really look forward to your application.
So we have a really exciting project proposal for this clinical PhD student entitled Mutations and Mosaicism in Human Embryonic Development. And this project was really born out of the idea that genes that are really important for early development are often particularly intolerant to variation. And when that's the case, mosaicism in the embryo can improve viability with variable developmental defects as a consequence. For example, ARID1A mutants have defects in their gastrulation. Knockouts fail to produce mesoderm, and they arrest at the stage of gastrulation. By contrast, the heterozygote dies mid to late gestation with neural tube and cardiac defects. By contrast, in humans, patients often present with intellectual disability, brain, cardiac, and skeletal defects. The complete loss of function heterozygous mutations are therefore more likely to be mosaic. So the project that we've proposed will use human gastroids to start to model some of these effects by using patient iPS cells and also genetically engineered human embryonic stem cells to model this early stage of gastrulation or perigastrulation development, which is the stage at which mosaic embryos will start to display features that are variable across the body plan. So the way that we make these gastroids is we take pluripotent stem cells in culture, we dissociate them into single cells, and we plate a defined number into a round-bottomed 96-well plate. And as a consequence, we end up with these organoid-like, what we call gastruloids, that undergo axial elongation, have patterns, gene expression, and representations from all three germ layers. So the plan with this project is to make these sorts of gastruloids from both patient iPS cell lines and genetically engineered pluripotent stem cell lines in different combinations, so that we end up with effectively an in vitro mosaic model of early development in the lab. So the research questions that this project is going to seek to answer involves how chromatin remodeling, including ARID1A, affects human gastrulation in specific. And how do patient-derived iPSCs show gastrulation defects in vitro? This will lead on to questions about how cellular mosaicism can affect developmental outcomes. And along the course of the clinical PhD, we'll provide ample techniques and training involving cell reprogramming, different imaging techniques, some sequencing, analysis and experimentation, as well as, of course, tissue culture techniques with pluripotent stem cells and these 3D in vitro model systems. So as we've mentioned, the team is a brilliant selection of PIs who are all have immense expertise in these areas, and the project will be held at the Francis Crick Institute, which has got a wealth of expertise and equipment and training opportunities. And so we think this is a really exciting project for somebody to get involved with. Thank you.